Knock, can I ask you a quick question? When I whenever somebody calls in, it puts their picture, even if they don't have video. Is there a way that I can get my picture to be the big one? Do I have to click here somehow? Like if I turn it off, then that's it, it's not there. If I turn it back on, it goes to me, but then as soon as somebody else calls in, then it goes to them. See? Now, it's them.
Let me see.
It makes me disappear if you, but it doesn't. That makes it big. But it doesn't
There should
let me
be
call.
a up arrow somewhere.
I guess it's. Change presenter. No. View video thumbnails.
No, that's not.
It's just a different way of seeing them.
See this down arrow
Yeah,
here?
it's.
There should
not-
be
it
an up
doesn't-
arrow.
Yeah. Anyway, it's not important. So. No, I don't want to make her the presenter. I'll just start. I found that happened last time and I didn't know how to change it.
There should be a. Is your session started already or no?
Yeah,
No.
I had it scheduled for noon. Okay, hello Greg and hello Siobhan. We'll get started. I'm just going to open it up to any questions that you guys have, and then if there's anything else I feel needs to be covered, I can go over that.
Vaughn, I'll let you start. I have a couple, so if you want to jump in first, I don't mind.
No, go ahead, because I got to get myself together here.
Okay.
All right. So, Professor Olkin, the first short essay topic, you're asked about bacterial growth as normal microbiota.
Yeah.
I understand the replication process and biofilm development and everything like that, but is there something specific you're looking for in terms of normal microbiota and not necessarily pathogens?
For this, I was really thinking of more the human body, and both pathogens and normal microbiota grow in the human body. So I was thinking of the example we had for the homework where the human body can act as a chemostat. And really anywhere in the human body, it's especially true in the gut, but really anywhere that bacteria would grow on or in the body, they have to have those essential elements or factors present for growth. So for example, it has to be like within the temperature range that the bacteria can grow in. There has to be food available and water available, and the pH has to be within a range that the bacteria can survive, and the enzymes, the metabolism can continue, as well as the salt concentration and things like that. So all of those growth conditions are the same, whether it's a pathogen or normal microbiota. And it's the human body that is, as far as human pathogens and normal microbiota of the human body are concerned, the human body provides all of those factors that the bacteria need to survive. And a chemostat is, or the gut is an example of that. The bacteria in the gut, whether they're pathogenic bacteria making us sick or whether they're normal microbiota. Does that clarify? let's see. Did we lose Greg?
Oh, maybe we did. I don't see him anymore.
Yeah.
I'll probably go back.
Okay. Well, hopefully that clarifies the first.
Okay. It did for me because it's more about the growth conditions, basically. For
Yes. So the
for any
focus of
kind
chapter 6 and what we learned about for the requirements for growth. Later we'll learn more details about normal microbiota, and we've already learned in chapter 2 the difference between a pathogen and a non-pathogenic bacteria, and as far as pathogens are capable of causing damage to the host, whereas non-pathogenic don't. And that's true of normal microbiota. The only exception is when they get in the wrong place. So
Right.
their normal microbiota are not everywhere in our body. They're only in areas that are exposed to the outside environment, like our entire GI tract. But, you know, if we have bacteria in our heart, they're not normally supposed to be there, and so then you'll get an infection, a serious infection, or a blood sepsis, life-threatening infection, because we don't normally have bacteria, and we're not supposed to have bacteria in our blood. That would be
Right.
serious. Yeah.
Okay. I mean, I know this is a pretty broad thing, but I've always had a problem with respiration and fermentation.
Okay, yeah. Hopefully this sounds familiar from what you learned in A&P or the general biology course that you took as a prerequisite for this course. And I'm not spending a lot of time on it, but I did want you to get kind of the big picture of the major differences and the kind of energy costs or energy yields that would be different depending on whether the bacteria are growing using respiration or fermentation,
Right.
and the environmental conditions. So just for bacteria, they're so diverse. They can grow, especially facultative anaerobes. They're very versatile as far as they can grow using respiration, or they can switch if the environment changes to be able to grow without oxygen. But for respiration, I want you to know the processes that are involved in that. So what are the really 3 main processes that are involved in respiration?
Well, there's ATP.
Okay, but I'm asking you for the pathways that are involved. So just let's back up a minute. Respiration and fermentation have something in common. What's that?
Well, it's energy.
It's all about making energy, making ATP, which without ATP,
Nothing.
there's no
happens.
life, right?
Right.
There's no metabolism, right? So it's that part of the metabolism that makes the energy for the other part of the metabolism, which is the anabolism, all the stuff that the cell has to build in order to stay alive. So that's the big picture. They really fall under the same umbrella because they're both catabolism. They're both concerned with making energy.
Okay.
And as we talked about in class, there's some similarities between the pathways, the actual series of chemical reactions that occur in these processes, and their differences. So when I'm talking about respiration, what are the 3 main steps that include all of these chemical reactions that are occurring in order to achieve respiration?
Okay, so what are the 3 main steps? And then for fermentation, you're asking a lot about the products of it.
Yeah, and I just want you to also understand the steps as well.
Okay.
So that clearly the products are different in fermentation than in respiration, right? So if the bacteria are consuming their favorite food, which is a simple sugar of glucose,
Yes.
what are they getting for a product out of that when you have respiration?
You're getting oxygen.
Oh, you're using oxygen.
Using
What are you
oxygen.
getting in the end?
Well, ATP.
ATP, and what are the products? You've started with glucose and oxygen. What are the chemicals that are on the other side, like as far as your reactants are glucose and oxygen, and you're getting products from the kind of rearrangement of bonds in those
Right.
molecules, right?
Yes. I don't know the.
So one way to remember it easily is that is carbon, right? So you have to think about carbon when you're thinking about catabolism. Catabolism is a breakdown of carbon-containing molecules. Glucose being 6 carbons, it's fully broken down to CO2 gas, which CO2, each molecule has just 1, right?
Okay.
So, but the key about catabolism is we're not just burning the glucose and completely oxidizing it down to its carbon and all the elements. We're doing it through, whether it's our human cells or bacteria, they're doing these processes through a series of controlled steps of slowly breaking it down piece by piece so that we can capture the energy that's useful. Right? If we just burned the glucose, a lot of energy would be released, but it would be released as heat, which is
Right.
useless to
Okay.
the cell,
Okay.
right?
Yes.
So that kind of big picture idea is what I want you to remember, not, what's step 3 of glycolysis or whatever.
Okay. All right.
But you should know the 3 major steps involved in respiration, which one is common between respiration and fermentation, and then how fermentation differs as far as the products. So when we're talking about glucose and oxygen, the glucose being fully broken down to a 1 carbon molecule of CO2, but 6 of those because you started with 6 carbons in the 1 molecule of glucose, right? So it has to be balanced. And then the oxygen is being converted to water. So it's taking those electrons that are passed from these energy carriers that are made along the way, along these many, many steps of respiration, and combining hydrogen with that oxygen. Because the oxygen, if it just keeps the electrons, is very reactive and very dangerous for the cell. It could actually kill the cell. That's when, getting back to another topic that's on this test is the oxygen requirements for growth.
Yes.
And if bacteria are incapable of breaking down these reactive oxygen species that build up in the cell, not because of respiration, but just naturally because of other chemical reactions happening in the cell, then
Are you
it will
talking
kill
about
them.
the ROS or something?
ROS, yeah.
Yes. Okay.
That's what we call the reactive oxygen species. So the cell has a way of just the protein, that's the last protein in the electron transport chain.
Yes.
And I should be showing you a picture so this makes more sense. Let's see, that is chapter 7 mostly. So that last protein in the electron transport chain is what holds the oxygen and doesn't release it until the hydrogen is combined with it to form water, which is harmless to the cell, right? It's like
Okay,
most of
got
the
it.
cell is water, right?
Yeah.
So that's the important step. So we have an overview. Where's the overview? Yeah, so they're basically, I would like you to know there's glycolysis, there's the Krebs cycle, and there's the electron transport chain that are all involved in respiration,
Yes.
right? And in the case of bacteria, they have a special site because there's no mitochondria. So the electron transport chain is found in the plasma membrane, and the electron transport chain, as we talked about in lab, it makes this imbalance of protons across the membrane. So there's a lot more hydrogen ions as the electrons are transported down the chain, a lot more hydrogen ions get pumped out to that periplasm, the outside of the cytoplasm, versus inside the cytoplasm. But then when those, let's see, I think I have figures somewhere here. Then when those are returned across the membrane through ATP synthase, I think the picture is in, yeah, here it is. Okay.
So that's what makes the rotation of the flagella.
Yeah,
Yep.
things like that. All of those, that imbalance of protons across the membrane, the only membrane that's separating the cytoplasm from the outside environment, is what drives this ATP synthase enzyme to make ATP, the 3-phosphate molecule that's high in energy, readily gives up energy, the energy currency of the cell that drives those unfavorable chemical reactions that are involved in building the parts of the cell and many other chemical reactions that are part of the metabolism of the cell. From ADP, which only has 2, it's more stable, it has 2 phosphates. So the key thing about ATP as the energy currency is that it's kind of it's constantly recycled. It's used up, converted, and releases its energy as it's converting to ADP. That release of that phosphate, which is held to the ATP in a very unstable bond, but a very high energy bond, so that when that bond is broken, it releases the energy that drives reactions that end up with having a product be much higher energy state, like building molecules. The product, like if you're building a sugar, is more complicated than the initial building block. So you can think of the macromolecules in the cell. The proteins need to be built from amino acids.
Right.
The nucleotides are put together to make nucleic acids. And so all of those reactions are going to be the ones that are going to need this energy from ATP. But then once the ATP is used up, it sits around as ADP. And so there's never really a separation. We assign a separation between the metabolism of the cell and call part of it catabolism and part of it anabolism, just to kind of categorize things in our head, but really it's all happening at once. Because you can never stop the production of ATP through these catabolic reactions, or the cell would be dead. It just would run out of energy to drive its metabolism.
Okay.
So it doesn't show the details here. Do I have one? Here's the overall reaction
Yes.
of the sugar, which is C6H12O6, the glucose, combining with water and oxygen to form water and CO2.
Right.
And let's see, for respiration there's aerobic, which this describes. This is aerobic respiration. In anaerobic respiration, it still uses all of these steps as far as the glycolysis, the transition step, the Krebs or the TCA cycle, the electron transport
But it
system.
just uses something else to grab that last electron, right?
Right, so rather than oxygen being the terminal electron acceptor that grabs onto that last electron, it's other molecules, like it can be sulfur or nitrate.
Okay, yeah, I did know that.
Nitrogen, yeah.
Okay,
So
cool.
here, we can see it a little more detail. Here is the electron transport chain. And you can see the last step is where you have, so they don't show physically in the lipid bilayer, but these are proteins that the electron transport system has. And they're just showing what the proteins do. So each arrow Black arrow is the protein pumping protons out
Yeah.
from the cytoplasm to the periplasm, as the electrons from these energy carriers, which are things like NADH is carrying electrons, FADH2 is carrying electrons. Those electron carrier molecules are unstable in their electron carrying state, and so they readily give up the electrons, and importantly are recycled, right? So they can be reused again in glycolysis because they can then accept more electrons. So they constantly cycle between their reduced form and their oxidized form,
Got it.
electron carrying form and their missing electron form. So, but the protons, at the same time as the electrons are being transported through the system, the protons are being pumped by the proteins that grab onto these electrons.
And that is what creates the proton motive force.
Right, so all this imbalance of proton concentration being greater outside than inside the cell, that's just a name for a proton motive force. Proton motive force is kind of like a fancy name for it. It's really just a gradient
Okay.
of protons.
Great.
But that gradient is really useful to the cell. It's a very powerful way to store energy that can then, once they go down their gradient, which is just a natural thing, doesn't require any energy, right? So molecules moving down their gradient is all passive movement, doesn't require any energy. So they readily go down their gradient trying to reach equilibrium, but because they're charged, they can't just go across the membrane down their gradient. They have to go through a protein channel. And ATP synthase and various other things like parts of the flagella motor will serve as this channel through which they can go, and that rush of protons down the gradient creates the energy necessary to make ATP, which is a very unfavorable energetic reaction because ATP has a lot more energy than ADP. So it's that proton motive force that's essential for adding inorganic phosphate to ADP to create ATP.
Okay. That was
Right.
a good explanation for me. Thank you.
Okay.
Okay. I don't think I have any other questions.
Oh, okay, great. So,
Oh, the three
we
main
didn't really get to the fermentation part of that.
Yeah, I know.
I kind of halfway answered
Okay.
the question, so let's look back at fermentation. So we know that there's some similarities. They're both catabolic reactions, right? But what's different about, so there are a few key differences that I want you to remember. And let's make this look bigger so I can actually show it to you. So in table 5.5, it kind of summarizes nicely. You've got aerobic, anaerobic. They're giving you a lot of ATP per one glucose molecule, per that 6 carbon molecule, right? They're giving in bacteria, because they don't have to shuttle the ATP out of the mitochondria into the cytoplasm. It's just directly made in the cytoplasm. You get theoretically 38 ATP per one glucose that's been consumed in that process and broken down completely, remember, to 1 carbon molecule of CO2. And you're almost there with anaerobic respiration. It's not the electron, terminal electron acceptors, those molecules that take that final electron, whether it's the nitrate or a sulfate, whatever it is not quite as a powerful electron sink as oxygen, so you don't get quite as many ATP or quite as many protons being shuttled across, and then not quite as much ATP made in the end. but it's pretty close. So you still got 30-something ATP molecules. And way more than with fermentation, where you're just getting 2. So where do the 2 ATP molecules that fermentation gets from catabolism? Where does that come from?
Where does...
What pathway is common between respiration and fermentation that produces a net gain of 2 ATP?
Why am I thinking glycolysis? Yeah, I'm looking at
Yeah.
that table and still not able to answer you.
Right, so it shows it here. So fermentation, the overall process, starts with breaking down glucose to
Yeah.
pyruvic acid,
Yes.
or it can kind of bisphosphoglycerate can also go into fermentative pathways. But let's just, for the sake of simplicity, just remember that glycolysis is happening. Glucose is going from a 6-carbon to a 3-carbon molecule, 2 3-carbon molecules of pyruvic acid, and then from there it's kind of, depending on what kind of bacteria or whether you're talking about yeast, it has its own fermentative pathway, whether it's alcohol fermentation or acid fermentation or mixed acid fermentation. There are a variety of fermentation pathways that occur after that one common process of glycolysis. But see here, glycolysis, actually, it makes 4 ATPs, but because the first step requires 2 ATP, you get a net of 2 ATP gained by the cell. So in the end, when you're doing the counting, it really only makes 2 ATP net gain for the cell to use in anabolism. So that's where the 2 ATP of fermentation are gained. The actual fermentative pathways of alcohol fermentation or acid fermentation, they don't make any energy at all.
Right.
So why are they important? Well, some of their products are important for us in producing food and alcoholic beverages and dairy products, etc. But also, without the rest of these processes, what do you see highlighted in blue here?
The NADH?
Right, so the NADH that's produced in glycolysis is being recycled back to NAD positive, NAD plus. Right? Without that step happening, the cell's metabolism would just stop.
Okay.
So that's a really important part of fermentation, is that it recycles the NADH so that it can be used again in glycolysis as an electronic carrier. All right?
Yes, thank you.
So, and you know, depending on whether you're talking about E. coli produces a variety of things, all highlighted here in this kind of reddish color. Yeast makes ethanol. Other kinds of bacteria, like Clostridium, make other alcohol as well, but a different kind of alcohol, an isopropanol. It's not used in making alcoholic beverages. Also chemicals like acetone and acids like butyric acid, butanediol. You know, all of these have, the importance of these being different products is that they can be, we'll see when we do, in 2 weeks when we do the biochemical lab, we'll see that these are important differences that we can use to distinguish bacteria that kind of look the same and might even grow similarly on media tests, but when you actually look at which pathway of fermentation they're using, you can distinguish. So you can do a test for mixed acid fermentation. And if your mixed acid fermentation test is positive, then, oh, okay, well, I probably have E. coli or only a handful of bacteria, but I definitely don't have Clostridium, or that maybe a more relevant comparison would be another gram-negative bacteria, Klebsiella, because Clostridium you can just tell right away it's not E. coli because you do a gram stain and it comes out positive. But for Klebsiella, E. coli and Klebsiella are really similar. They have the same gram stain characteristics of gram-negative. They both look like rods or bacilli, and they both grow similarly on the media tests. So it's really not until you get to this biochemical test looking at the products, the pathway that it uses for fermentation, whether it's the butanediol pathway or whether it's the mixed acid fermentation pathway, that you can tell the difference between them.
Got it.
So that I'm kind of getting ahead of myself, but that's why I want you to kind of remember some of these differences in the fermentation products. Because that later we'll learn about how relevant they are to identifying a species of bacteria that's a pathogen, for example.
Great, thank you.
Yep. Let's see. Let's see, did we lose? All I see is you, Siobhan. Let me see, stop sharing. I thought
There are
we had a couple
more
other people.
calls.
Oh yeah, we have a couple of, oh yeah. So anyone else have a question that they want to, ask now that I've finished that one? Ulrich, you're muted. Ulrich?
Yeah, I'm actually outside.
Oh, okay.
That's why. Yeah.
That's fine. I'll mute you again. But if you do have a question, just remember, unmute so I can hear you. Anyone else have a question? All I see is call-in user, so I don't know your names.
Professor Olkin, this is Greg again. Sorry, I just got
Okay,
to,
Greg,
on the
great.
work.
Yeah.
I was going to ask you in the question about discuss disinfection and chemical control of bacteria. What are you looking for in terms of chemical control? And if you already covered this while I was gone, please feel free to tell me to watch the...
Nope, we didn't cover it. So, let's see. Discuss the effectiveness of disinfectants? Which?
No, I was looking at the chemical control of bacteria. What,
okay. For the essay, the open response. Yeah, so for this, so this is two things. We did the experiment in lab, and we just finished reading chapter 13, the first part, where it talks about physical and chemical means of controlling bacteria. So the chemical means would be the relevant chemicals that I'd really expect you to remember are the ones that we tested in lab, like hydrogen peroxide, alcohol, and bleach, which is a halogen-containing chemical. And that all chemicals have different levels of control. There are 3 different levels of control. There's high, intermediate, and low, depending on what, how much of how stringent it is, or what amount of disinfection you can get. So low-level disinfection is something like we saw with alcohols affecting some bacteria in the assay but not others, and halogens also. But then when you're getting to the high level of disinfection, we're talking about chemicals that are capable of sterilizing. So completely removing all of the all of the microbes that are present, including the more challenging ones like bacterial endospores. So really there's no chemical that can get rid of high levels of bacterial endospores, but something like a gaseous form of ethylene oxide, like your book gives as an example, is a high level, it can achieve high level disinfection. Disinfection being the removal or the elimination of microorganisms, and really what we're talking about is pathogenic microorganisms, because those are the ones we're concerned with when we use disinfection, from a surface, right? So disinfection is a surface, it's not from your body or from a living tissue. That term is antisepsis, or and you can never sterilize any part of your body. You would have to kill your own cells to do that, and that's not practical. So antisepsis is not sterilization, it's just reducing the number of microorganisms to a safe level. But when we're talking about disinfection, I'm talking about the chemical control, and the chemicals I'm talking about are the ones we saw, for example, in the lab experiment we did. So you should know about how do they work. They have three main targets in the cell. Something like an alcohol, like isopropanol or ethanol, will be destroying the membranes. So remember the third step of gram staining, where you use the decolorizer. That's destroying the outer membrane of the gram negative, as well as the inner membrane and the inner membrane of the gram positive, right? So that's basically how it kills the cell, and it's capable of killing gram positive and gram negative. We just see a difference in the case of the gram stain because it's dehydrating that cell wall that traps the color in the gram positive but not in the gram negative. But in the case of disinfection, ethanol or alcohols are low-level disinfection, are capable of low-level disinfection, and they're targeting, the part of the cell that they're targeting is the membranes. They're destroying the membranes. Without a membrane, there's no way for the cell to kind of keep what's inside the cell inside and what's outside outside, right? And then the other, where most chemicals, if not all, and I think most of them, are also effective at oxidizing or clumping and aggregating proteins. And remember, proteins is like the bulk. Of course, water is the majority of what's in our cell, but if you look at the macromolecules, protein is the bulk of what's in there. And those proteins are mostly enzymes that are necessary to carry out the metabolism and keep the cell alive, right? So without those proteins able to act because they're kind of stuck together or they're modified by oxidation, then that's it, the cell is dead. So, and then finally, high-level disinfection is achieved by things like formaldehyde. Aldehydes target the DNA, and without that genetic information, the cell can't survive. So those are kind of the three main targets in the of chemicals in their role in disinfection. Any other questions about that, or did that clarify for you? No, I think did we lose them? Oh, we have someone joining. Hi everyone, we're in between questions, so does anyone else have a question that they want to ask about the from the study guide or any of the material you're responsible for the exam this week? So I'll just remind you from the study guide. Oh wait, first I have to share with you. Okay. So just a reminder from the study guide, you'll need to be sure that you're able to label a growth curve. So those 4 phases of growth, lag, log, stationary, and death. Be sure that you're familiar with the steps of biofilm development and their relevance in medicine and their challenge the challenge to treating them when they're biofilm infections. Let's see. Be sure you know the difference between respiration and fermentation. In general, physical methods and how they do, especially the autoclave, because we use that so routinely to sterilize and i.e. completely remove the presence of any microbes on tools, for example, surgical tools. Let's see, what else? You should also know the oxygen requirements. I think I have that somewhere here. Yeah, so all of the variety of terms used for describing the oxygen requirements of bacteria. Some have to live in an environment that contains oxygen, so in the air. Others are killed by that environment. So aerobes, anaerobes, facultative anaerobes, microaerophiles, all of that, and be able to understand where bacteria are capable, that are, for example, anaerobes are capable of growing. So they would be killed in the presence of oxygen. So if I asked you to label a culture tube that had agar, they wouldn't be able to grow in the area where the air is present, but they would be able to grow far away from that interface with the outside air. All right, any other questions? I'm not sure who called in, but Ulrich, do you have any questions? Or Siobhan, I think she's muted, so I'm not sure if she's listening.
I'm good. I'm just hanging on to see if there's anything else.
Yep. Okay, if there are no more questions, then I'll end the session.
Thank you so much for your time.
Okay, thank you.